Any questions, guys, before we start? Anything you specifically want me to go over or anything like that? No? So what I'm going to do, Melissa, you have something or?
No, I don't have anything.
Okay. What I'm going to do is I'm just going to go over the quizzes for chapters 1 through 4. I'm going to go over the homework. And then if there's any other questions that sort of are prompted by that, that's fine. So it's just kind of just going to be a review of those chapters. Remember that last week, basically we moved on chapters, well, 4 was part of last week too, I guess, but chapters 5 and 6, and then 7 and 8, I will put up in the next couple days. Okay. So then that gets you to exam 2. Exam 2 is chapters 5 through 8. Okay. So today I will make exam 1 available around 1 o'clock today. You have until the end of the day, 11:59 p.m. on Friday to complete exam 1. Okay. So exam 1 is chapters 1 through 4. That's what I'm going to review today. Anyone has questions, please let me know. Like I said, as I'm kind of going through, interrupt me if there's something you want me to go over. All right. I think that's pretty much it. On Thursday, I will go over the labs, right? Those sort of labs from the first, I guess it's modules 1 through 3, and then modules 4 through 6. Those labs are due by Thursday. And then that's what will be on your first lab quiz. Okay. So I'll talk about that Thursday. And remember, the lab exam really is just a lab quiz. It'll be 10 to 20 questions, all multiple choice, right? It's just meant to just be sort of just to kind of recap and just an additional assessment. But really, most of your lab work is doing those reports and those assignments. All right, guys, any questions? Otherwise, I'm going to start off by going over the chapter 1-2 quiz, then the chapter 3-4 quiz, and then I will go over the homework for chapters 1 through 4. Any questions? A couple people came on. No? Okay, so let me do that. Let me share my screen. I'll start off, like I said, with that first quiz. And again, the quizzes are really, not meant to be anything crazy. They're usually pretty simple and straightforward, just to get you guys to review the material. So if we look here at this first question, again, first quiz is only 10 questions. Human forces that destroy the tropical rainforest lead to an increase in biodiversity. So what's that, true or false? Does it increase biodiversity or decrease biodiversity?
That is false.
It's false, right? So in terms of biodiversity, right, basically think of that as the number of species that exist, and also sort of the number of genes within the species, and sort of the amount of genetic diversity and just species diversity that exists in a given ecosystem, right? And so anyway, it's false, okay? It would decrease. All right. So next question, and so like I said, stop me if something doesn't make sense. Atomic number is equal to what? The number of protons and neutrons, the number of neutrons, the number of electrons, or the number of protons? Which one is that one for atomic number? Well, what's the number of protons and neutrons? That's the atomic mass, right? So atomic number is what?
Number of protons.
Number of protons, right? Thanks. And then, so if you know the atomic mass, right, and you know the atomic number, you can easily figure out the number of neutrons, okay? And how come we don't talk about the number of electrons when we talk about atomic number or atomic mass? Because electrons, their mass is negligible, right? So when we talk about the mass of an atom or the atomic mass, it has to do with protons and neutrons. Those electrons float along the outside, right? They're super important for the chemical behavior or chemical activity of the atom, but they don't contribute to their mass, okay? Just a reminder. In terms of charge, right? Protons are positively charged, electrons are negatively charged, and neutrons are neutral, okay? No charge. All right, so like I said, if there is questions on anything, just interrupt me and ask me to elaborate. So I'm sort of obviously using this as our sort of means of review, okay? All right, so next one, the outermost electron shell of an atom is called the valence shell, right? And so remember, that's what for our purposes, that's what we're focusing on, right? Because it is the number of electrons in that valence shell that, again, dictates the behavior of that atom, okay? In terms of what it's going to form bonds with, how it's going to form bonds, and what have you. All right. Which of the following includes prokaryotic organisms, right? So in which of these domains do we find prokaryotic organisms? And so for this one, right, the answer is both archaea and bacteria. Remember, domain eukarya is all eukaryotes. And remember, domain eukarya is then broken up into different kingdoms, right? Phylums, different families, orders, right? Different species all exist in domain eukarya. Domain bacteria is just, there are no kingdoms, though that may change, but right now it's really just all the different bacterial species are part of that domain, okay? Archaea, same kind of thing. All right, so that one. Okay, unequal sharing of electrons is characteristic of what type of bond? Okay, so if we're talking about sharing, it's a covalent bond, right? And if we're talking about unequal sharing, it's going to be polar, right? Non-polar is equal sharing. Polar is, sorry, unequal sharing. For a little while. When we, hydrogen bonds, right? When we talked about water and sort of the formation of hydrogen bonds between water molecules, we talked about that a lot. It has to do with the polar nature of those water molecules, right? Remember having, you know, some of the part of the molecule having a partial negative charge versus a partial positive charge, okay? And ionic bonds, we you know, that our example was with sodium chloride, right? So that's sort of usually in non-organic molecules we see ionic bonds, okay? But the answer to this is polar covalent bond, okay? All right, so water is a polar molecule. The answer is yes, you should know that, and you should know why we consider it a polar molecule, okay? Remember, and I'll probably, I mean, I can pull up, you know, like a slide from chapter 1. Is that 1 or chapter 2? Regardless, I can pull up a slide if somebody wants clarification. So if somebody says, look, I want you to clarify the sort of polar nature of water or whatever, I'll pull up a slide. That way we have a figure to go over that a little bit more. All right, so for right now, polar unequal sharing, that molecule is not the same on both sides, right? It's different because of that unequal sharing of electrons, and that's what makes it a polar molecule. All right, so there's lots more to say about that, the polar nature of water and hydrogen bonding and all that good stuff. And so I talk a lot about that in that recorded lecture. All right, a solution with a pH of 9 is what? Acidic, basic, or neutral, right? The answer is basic. So 7 is neutral, pH 7 is neutral. Anything below 7 is acidic. Anything above 7 is basic. All right, the level of organization that includes cells of similar structure and function is what? It's a tissue, right? And so if we talk about, remember we talked about those biological levels of organization, and we started with most simple and then moved to more complex, right? So most simple being atom, right? Then molecule, then cells, then tissues, right? Organs, organ systems, and we went all the way up to biosphere. Okay, so make sure you know those levels of organization for the exam. Any questions, guys, so far? All right. Okay, which of the categories listed below is most inclusive? Okay, so remember that terminology when we're talking about most inclusive or least inclusive, meaning which group is the largest or contains the most, the largest number of species underneath it, right? So domain, right, we talked is the largest category, okay? All of these fit underneath domain. Also, kingdom fits underneath domain, but it's not listed here. Okay, so all of these categories here are less inclusive. Okay, and they're more specific, I guess, if you want to think about it that way. All right. Okay, low levels of radiation are used in PET scans to detect brain activity. Right, so the answer is true there. We also talked about another type of imaging technique, right, where basically iodine was labeled, right, and you could basically image the thyroid gland, okay? But those are, in that case, basically the patient is being injected with a radio-labeled molecule, okay? So it has to be low levels. All right, and then remember there were also, we gave examples of high uses of high levels of radioactivity, right? So those are all things to, again, be familiar with. All right, so hopefully we got them all right. Okay, so that was the quiz on chapters 1 and 2. So is there anything in chapters 1 and 2 that you would like me to go over a little bit more? I mean, again, all of it is in the recorded lectures, and I think I make a point to sort of point out what I think is sort of most important and things that I'll emphasize on the exam. So again, I can't reiterate enough, if you don't listen to the recorded lectures, you're going to be at a severe disadvantage, right? Just looking at the slides isn't enough. All right, so what I'm going to do is I'm going to go over, well, so one thing too, let me just pull this up. Just, and also there was a couple people that had a question about this. So the exam 1 review sheet, so let me just make sure I address this. And it's also probably good if you have yours out while we're sort of going over things. So for the review sheet, remember, all this is just an outline, right, of basically what I went over in all of those recorded lectures. For the assignment to complete this, basically you are filling in, you know, the sheet. It might be, if you're going to do it electronically, probably best to maybe put it in a different color font or whatever. You are either defining, you're providing examples, you're explaining some of these topics. There's no really right or wrong way of doing this. I had a couple people ask me, you know, how are you supposed to do it? It's just an outline and you're filling in your notes. It's just provides you with a little bit of framework so that when you listen to the recorded lectures, you have some sort of active, you know, or activity going on while listening, okay? So it's a very, it's active learning in its sort of most minimal, I guess, sense, right? But it's something that's helpful. In class, we actually have them out in front of us, and I usually have people manually writing on them, and I check them as we move around just to see how people understand. So anyway, that's clarification on that, all right? So we just went over the quiz for chapters 1 and chapter 2, okay? So remember, chapter 1 was really just an introduction. Probably a lot of the stuff that's in there you probably know about or heard about. The one thing that I didn't really, I guess, emphasize on the quiz was the scientific method. So make sure we sort of understand the basics of that. If I showed you a graph or something like that, we should be able to interpret the graph, but it would be kind of, you know, simple questions like that. For the basic chemistry chapter, we just sort of went over a couple things actually from that. So make sure, again, we understand all the topics in there as well, okay? We talked a lot about the chemistry of water, so hopefully you guys got that. That's probably an important thing to study. All right, so like, for instance, if you're writing in some notes here about the chemistry of water, probably a good idea to draw the structure of water, right? Show which part is electronegative or which part has a partial positive charge, which part has a partial negative charge, okay? Those are things that should be written in your notes there. All right, so moving on to chapters 3 and 4, I'm gonna go over that quiz. Actually, you know what? Let me go over the homework to chapters 1 and 2 so that we kind of close that off, and then I'll go over the quiz and the homework for chapters 3 and 4. All right, so just to remind you, 3, right? Chemistry of organic molecules. We talked about all the different types of biomolecules, talked about their function, gave examples, talked about reactions that occur in their synthesis and also in their breakdown. So Once you get to chapter 3 and 4, things start to get a little more detailed. All right, so let me just minimize that for now. And let me go back. What did I do now? 1 second, let me move this out of my way. All right, and let me just go back now. Like I said, I just want to make sure I go over chapter 1 and 2 homework in case anyone has a question. Okay, so common characteristics of life, right? I mean, I'm not going to go through and list all these. I mean, you know, I can pull up the PowerPoint. Actually, let me just do that real quick just to kind of point out where these things are. But I purposely don't post answers to the homework because all of these homework questions are so straightforward. It's about just going back to the lecture and writing down those answers. If I post answers, then people don't go back and look on their own. So I want you guys to do that. If you have a question about it, ask me, right? So let me just quickly pull up. I think that's the easiest way to do it because again, I'm not going to write out the answers, but let me just point out where they are because they are so straightforward. So it'll take me 2 seconds just to open that up. And let me just pull up chapter 2 as well. And then I can just toggle back between the lecture and that. Sorry, I should have downloaded that before we started. That's my fault. Okay, and then let's do. Okay, all right, go away. Why? All right, so let me just go back to that assignment. Okay, so the first one, common characteristics of life. And again, if I ask you a question like this on the exam, I expect that the answer comes from our lecture or my recorded lecture and that it's not something that's Googled and then just, you know, because obviously you can answer that in very different, in a bunch of different ways. But any question that I do ask you in terms of essay questions on the exam, I'm expecting that the answers come from our notes, right? Our book, our lectures, what have you. If they do come from Google, I can tell, and I'll give you guys a 0 for that question. So I hate to have to say that, but I have to say that, okay? So that just reminded me of that. Okay, so common characteristics of life. If we, what happened to my, sorry, I'm trying to get my picture back there. Yeah, there we go. Okay. All right, so common characteristics of life, we went through them, okay? Right at the beginning of the chapter, we talked about living things being organized. And then the living things being organized then corresponded with those levels of biological organization, which I believe is that second question for the chapter here, okay? So again, these are all straightforward, listed at the beginning of chapter 1, all right? So levels of biological organization are here. And then, again, that first characteristic life is organized. And then we talked about life requires materials and energy. These are all listed and numbered, okay? Pretty straightforward. So if anyone has any questions about that, let me know. All right. Number 3, how does natural selection result in adaptation to the environment? And I think we might have kind of went over these also at one of our other earlier class meetings. You know, we talked, again, we didn't exhaust the topic of natural selection in this chapter. It was really meant just to be an introduction, but I want you guys just to understand the process in general, right? One of the things to point out is down here where it says mutations fuel natural selection, right? The idea is that there was a genetic change that occurred by chance that then provided the organism with some trait that made them more successful, right? That was beneficial to them. The environment, the natural part of it, then selects for that trait, okay? And so the adaptation is that trait that occurred because of that genetic change that, again, made that organism more suited to the environment. So you can kind of answer that in your own words in a couple different ways. And in fact, if I did ask you that question on the exam, I would expect you to try to answer it in your own words to show me that you understand that, okay? Does anyone have questions about that? Okay, So for chapter 2, remember that's our basic chemistry chapter. First question, what's an isotope? What's a radioactive isotope? And describe 2 uses. And again, we did go over these, I know, in an earlier class here. So I kind of just talked about this already on the quiz, right? Again, the answer to this question is in chapter 2, where we were talking about isotopes, okay? So generally, what's an isotope? An atom of the same element that differs in the number of neutrons, okay? So they have different atomic masses. You can see here, right? These are all isotopes, carbon isotopes. Some of these will spontaneously decay, right? And give off energy, and those are then radioactive isotopes, okay? So they're not stable. And then the next part of the question asks about uses of radioactivity, okay? And so we talked about low-level radiation in imaging techniques, and then we talked about high levels in terms of increasing the shelf life of fruit, and also in radiation therapy, okay? All right. For number 2, explain why water is a polar molecule. What does the polarity and shape of water have to do with its ability to form hydrogen bonds? Super important question and topic, okay? So let's move down to the part where we talk about water, okay? So if we look at this picture right here, right, this is showing water as a polar molecule, okay? Well, one thing, let me just point out, these molecules here, right, are non-polar, okay? Equal distribution of the electrons, as opposed to water, which is polar, okay? Again, if it was non-polar, the hydrogens would be on the same level of the oxygens, okay? They're not. And because there's unequal sharing, it creates this partial negative oxygen and the partial positive hydrogens. And so because of that, you have, it's different on one side of the molecule versus another, okay? That partial negative and partial positive charge gives water the ability to form these hydrogen bonds, okay? Because oxygen is partially negative, it then draws positive ions or other partially positive molecules towards it, okay? That's the hydrogen bond. So when you think about a whole bunch of water molecules, right, together, and I think I have a picture that shows this right here. Let me make this bigger, sorry. Because again, I think this is super important. So if you look at this picture here, right, guys, you can see, again, these are a whole bunch of water molecules together. And again, these are hydrogen bonds, these interactions between the partially negative oxygen and the partially positive hydrogens, okay? Again, this doesn't only have to occur between water molecules, it can occur between other types of molecules as well, okay? Also remember that hydrogen bonds, as one hydrogen bond, they are weak interactions, but when there are a bunch of hydrogen bonds, that then collectively becomes a strong interaction. Okay. All right, so it says, what does its polarity and shape have to do with its ability to form hydrogen bonds? I just went over that. All right, atomic number and mass number. I sort of went over that during the quiz. And again, this is more at the beginning of the chapter. We talked about this more, okay? So we should know atomic number. We should know what atomic mass is. If I show you something like this, we should know that 12 is the mass number or the atomic mass, and that 6 is the atomic number, right? And again, we should know atomic number is equal to the number of protons. Atomic mass is the number of protons and neutrons. All right? Any questions, guys? I mean, I think I hit almost all of the high points by going through the quizzes and the homework here. All right, so like I said, I'm going to move on to chapters 3 and 4. I'm going to go over the quiz first, and then I'll go over the homework as well. So let me go back. Let me make sure I also pull up those lectures just so that I can point out some of the highlights like I did for chapters 1 and 2. Oops, why did I do that? That was dumb. Okay. So let me just do that. Make sure I have these downloaded and ready to go. And chapter 4. All right. Sorry, well, I don't know why it's taking so long. There we go. All right. Okay, so let's go over that chapter 3-4 quiz first, okay? So let me pull that up. And that's not what I meant to do. Sorry. Need to go to preview. All right. Okay, so first question. So there are more questions on this quiz, right? So cellulose is an example of what? It is an example of a carbohydrate. It is a structural carbohydrate, right? And you find cellulose in plant cell walls, okay? There were other types of structural carbohydrates that we mentioned, peptidoglycan, chitin, okay? Those were some other examples. Chromatin. Where is chromatin found in the cell, right? Because remember, chapter 4 was parts of the cell or cellular organelles and their function. So we find chromatin in the nucleus, right? Chromatin is the term that describes DNA and proteins, right? So DNA is packaged by special proteins called histones, other proteins as well. And so again, chromatin is the term that describes DNA with its associated proteins. Okay, so we find DNA in the nucleus, chromatin in the nucleus. All right. Saturated fats have double bonds. True or false? False, right? What types of fats have double bonds? Unsaturated fats. Remember, the double bonds create space within that molecule. Okay, and so an unsaturated fat will have one or more double bonds. Saturated fats have all single bonds. All right, a protein that is destined to be secreted out of the cell will move from the rough endoplasmic reticulum. So this is that endomembrane system. It's one of the homework questions. To the what? Where will it go next, right? It goes from the endoplasmic reticulum to the Golgi apparatus, okay? And then it will be packaged in a vesicle and then be sent to the plasma membrane to be secreted out of the cell. So again, that's sort of following along that endomembrane system that I talked about. Okay. Nuclear envelope is composed of what? Nuclear pores, inner nuclear membrane, outer nuclear membrane, and basically all of these other three things. So the answer is that it contains all of the other components, okay? The pores, remember, are openings that allow proteins, things like RNA, right, to be transported into and out of the nucleus. All right, all the following are biomolecules except, so the 4 classes of biomolecules: proteins, lipids, nucleic acids, and carbohydrates. Salts are not biomolecules. Okay, monomers of proteins. I think I asked you guys this question like 50 times. The monomers of proteins are amino acids, right? Proteins are made up of amino acids. Remember that proteins are made up of amino acids, but then they also need to be properly folded before they function, okay? So really, it's polypeptides, right, that are made up of the amino acids, and then that polypeptide chain folds, right? Remember, we talked about levels of organization of proteins: primary, secondary, tertiary, and quaternary. So be familiar with those different levels and what's happening. But basically, it's sort of initial interactions, and then more folding sort of occurs. And then once it's in its final conformation, it then is a functioning protein. Okay. Prokaryotic cells do not have a membrane-bound nucleus. That is true. They also lack many other membrane-bound organelles. They are more simple than eukaryotes. Okay, they're smaller. There's some other differences as well that we can go through. There's a nice table that's in your presentation or PowerPoint that shows that. So definitely make sure you know the difference between prokaryotic cells and eukaryotic cells. All right, Golgi apparatus is part of the endomembrane system. True, I just kind of talked about that. Okay, remember that endomembrane system, basically all of these organelles are connected by membranes or vesicles. Okay, so that's why it's called that endomembrane system. Starts basically with the nuclear envelope, right? All of them are sort of continuous, just about continuous with one another. All right. Plants store glucose as what? Right, as starch. Okay, so when we think about complex carbohydrates, right, starch is one of them. Glycogen is also another one. So animals will store glucose as glycogen. Plants store glucose as starch. Okay. We mentioned cellulose before, right, which is a structural carbohydrate. Maltose is also a carbohydrate as well, a more simple carbohydrate. Okay, the synthesis of lipids occurs in which organelle listed? Right, so where does lipid synthesis occur in the cell? Okay, it's the smooth ER, right? Remember the rough ER is studded with ribosomes. Protein synthesis occurs on the ribosomes, right? And then once those proteins are made, they then can enter right into the ER there and then move through that endomembrane system and maybe be secreted out of the cell. Peroxisomes, what's the function of a peroxisome? Remember so those were kind of talked about separately. We talked about lysosomes in sort of as part of that endomembrane system because they are associated with the endomembrane system just because of the way they're derived. But lysosomes and peroxisomes in terms of function are very similar, right? They contain enzymes that will break down larger molecules or when you think about lysosomes, right? If something's coming in to the cell via endocytosis, like a bacteria or something like that, then will fuse with the lysosome, and then those enzymes that are inside the lysosome can break it down. With peroxisomes, there are characteristic molecules that get broken down by the peroxisome. The reason why they're called peroxisome, because the chemical reactions within the peroxisome actually form peroxide. And then that peroxide needs to then be broken down as well. Okay, but same idea. They're kind of, they contain enzymes that can break things down. Okay, things that the cell doesn't want. All right. Okay, and ribosome protein synthesis. All right, so that was just a review of some of the organelles there. All right, triglycerides are an example of what? What type of biomolecule are triglycerides? Right, they're lipids. What's another name for a triglyceride? Fats, right? And basically this name tells you about their, their structure. All right, the cell uses blank reaction to synthesize biomolecules, right? So remember we talked about hydrolysis and dehydration reactions, okay? So which one will synthesize a molecule, right? It's dehydration, right? Water is removed. Right? Red blood cells have a greater number of lysosomes than other cells in the body. So I just gave you an example about taking in a bacteria or something like that. It's not red blood cells that do that, right? It's white blood cells. So the answer is false. I didn't really meant to for that to be a trick question. Remember, white blood cells are your immune cells, right? And one specific type of white blood cell is a phagocytic cell that basically can take in, like I said, bacteria, then that will be broken down by the lysosome. Red blood cells function in carrying oxygen around the body, right? They're mostly membrane and cytoskeleton. Okay, blank cells have a cell wall. Plant cells, not animal cells, okay? Hopefully we know that. All of the carbohydrates listed below are monosaccharides, except which one? Right? It's glycogen. Remember, glycogen is very large, complex molecule, and it's a storage molecule, and essentially it's made up of a whole bunch of glucoses, okay? The production of ATP occurs in which organelle listed below? Mitochondria, right? Remember, mitochondria are the powerhouses of the cell. That is where ATP is produced. That's where cellular respiration occurs. That's our topic for chapter 8. So we're going to talk about the mitochondria a little bit more in that upcoming chapter. Okay, I think I already defined the function of these other organelles as well. So basically, you need to know the function of those major organelles that we talked about in chapter 4. Protein synthesis occurs in which organelle listed below? Again, I already talked about this. It is the ribosome. So, you know, if I asked you that and I had put rough ER as a choice, I wouldn't really do that because it's a little misleading. But remember, the ribosome is actually where the protein is made. The ribosomes just sit on that part of the ER, and that's why it's called the rough ER. Okay, so really, the rough proteins aren't synthesized in the rough ER, right? It's really on the ribosome. Okay, so protein synthesis is ribosome. Which part of a phospholipid is hydrophilic, right? So when we think about a phospholipid, it has a phosphate-containing head, right, and 2 fatty acid tails. And I'll, you know, pull up the chapter just to point that out as well. But which part of that is hydrophilic, right? It's that polar head. Okay, those fatty acid tails are hydrophobic, right? Which means they don't readily dissolve in water. They're sort of water hating, whereas hydrophilic, that part is going to readily dissolve in water. And that's why those heads face towards the outside of the cell and towards the inside, and the tails are in the middle. Okay. All right, last one. The blank structure of a protein is the linear sequence of amino acids or polypeptide chain. So I kind of talked about this a little bit earlier. If we talk about primary versus secondary versus tertiary versus quaternary, the primary structure of a protein is the polypeptide chain or the sequence of amino acids. Okay. Secondary is characterized by the formation of beta pleated sheets and alpha helices, and then in the tertiary, that's when you have that final folding of that polypeptide. If a protein has a quaternary structure, it is just because it has 2 or more polypeptide chains. Okay, that's the only instance where you will have that quaternary structure. All right. So that is that. So again, that allowed us to kind of review a lot of sort of important topics within those 2 chapters. All right, so let me now just go back to the homework questions for chapters 3 and 4. And then again, I'll sort of pull up the lecture to correspond with those questions. And that's pretty much it. The other thing to point out, guys, as I see this here, remember, there are animations in these folders that may be helpful. Also, this review packet just has some extra practice questions. Also a summary of the chapter. You may find that helpful as well. Okay, so those are always there in case you need them. So let's look at chapter 3 homework. First, I actually do have the answers posted there, but whatever. I still don't think it's useful. I can make them available, but I don't like to. All right, so let me minimize this and then pull up that chapter 3 lecture. Okay, so for the first question, describe the synthesis and degradation of one type of biomolecule. So you could choose any, right? If I asked you this on the exam too, and for your homework, you could have chosen any biomolecule, right? Again, when we talk about the synthesis and degradation of these biomolecules, they utilize the same general type of reaction, right? Remember we talked about dehydration and synthesis. So I'll just go with one here. If you want to specifically talk about carbohydrates, right? Here's an example of dehydration, right? So 2 glucose molecules, right? Or 2 monosaccharides joining together via a dehydration reaction to form maltose, right? Which is a disaccharide here. And then obviously water is lost because of that dehydration reaction. If you then go the other way, let's make it bigger. If we then go the other way, right? And we talk about degradation, you're starting with a disaccharide like maltose, right? 2 glucose molecules joined together there. And now what you're going to do is you're going to break this bond, right? You're going to break this via a hydrolysis reaction. Oops, I went the wrong way. And you're going to end up with those 2 glucose monomers. Okay, so you could have chose this. You could have chose proteins or whatever biomolecule you wanted to do. All right. So if we go back Number 2, name 2 types of polysaccharides and describe their function. I mean, I kind of already did that in the quiz, right? You could have named glycogen, starch, right? Cellulose, chitin, any of those. And so these were our examples of polysaccharides, okay? So any of those in terms of what their function is, okay? So remember, again, polysaccharides, complex carbohydrates, these are larger molecules, much more complex. All right, let's see what is next. Describe the difference between an unsaturated and a saturated fat. I also kind of already did that as well, but let's take a look at the pictures just to kind of point out. Yeah, I think this is probably the best slide that sort of shows that, right? 1, when we talk about fats or triglycerides, they're named triglycerides because you have 3 fatty acids joined to a glycerol, right? If we talk about unsaturated fat versus saturated fats, unsaturated fats contain double bonds, okay? So I could put this up on the exam and say, tell me whether this is unsaturated or saturated. We should be able to point that out. You can see, if you look at the space filling model here, that because of these double bonds, you have a lot more space between these fatty acids, as opposed to in that saturated fat where it's all single bonds here between the, between the carbons, right? These fatty acids are closely, tightly packed together. Okay. All right, what are the monomers that make up proteins? Super simple question. And the answer is amino acids, right? And what are the monomers that make up nucleic acids, right? So remember, nucleic acids, RNA and DNA. What are those monomers, right? They are nucleotides, okay? So wait, let's get to that just to kind of look at that again. So remember the levels of protein structure. This is a really nice diagram or figure that shows that: primary, secondary, tertiary, and then quaternary. Sorry. Okay, so if we talk about nucleotides, right, those are the monomers of RNA and DNA, okay? Remember, there are different types of nucleotides, okay? All right. Anything else you want me to go over from this chapter, guys? All right, so let's look at the homework for chapter 4, which I think was only like 2 questions. 1 second. And then we will be done. All right, so chapter 4 assignment is here. So describe the relationship between the components of the endomembrane system. And then the second question asks about the function of chloroplasts and mitochondria. Okay, so pretty straightforward. Let's look at that endomembrane system just to kind of review. And then we'll talk a little bit about chloroplasts and mitochondria, and then basically we will be finished. So for chapter 4, remember that this is cell structure and function. Basically, we're going through all of the different organelles of the eukaryotic cell and describing their function, right? And also talking about how they work together, especially that endomembrane system, okay? Right, and so I did point this out in the recorded lecture that though this looks like super busy and the words are really small, this is a nice slide to study from because it shows you where those organelles are located and also the function. Remember, on the lecture exam that that's going to be available today, I'm not going to ask you to label diagrams, but on the lab quiz, I will have a diagram up like this and ask you, I could say, all right, point to the mitochondria and ask you what that is, okay? But for the lecture exam, it's just knowing the function, all right? But we will do that in the lab quiz. Okay, so the one thing that I just wanted to kind of go over is that endomembrane system. I don't like that figure. Let me just see. I think there's a nice overview figure that I can pull up. Yeah, here. So if you guys look at this, so I think I pointed this out too in the recorded lecture, that I do like this figure because I think it shows you kind of everything at once here. So when we're talking about the endomembrane system, right? Remember, it basically starts with that nuclear envelope. And then you have the rough endoplasmic reticulum, right? So protein's going to be made on a ribosome that sits on that rough endoplasmic reticulum. It then can enter into the endoplasmic reticulum and then eventually will be transported, right, to the Golgi apparatus. Oh, sorry, we're going this way, right? So you can see it gets packaged into a vesicle. All of these things are associated with each other, either directly through the membranes or sort of indirectly by a vesicle. But basically, it's all composed of membrane, okay, and just space, essentially. So what this is showing you, if we're talking about a lipid that's synthesized in the smooth ER, right, it then will be transported to the Golgi also, right, via a vesicle, okay? Once it gets to the Golgi is going to sort and package all of this cargo, whether it's a protein, whether it's a lipid, and then again, package it in a vesicle. If we're talking about a secreted protein, it then will be sent to that plasma membrane and secreted out of the cell, okay? So that's all kind of following through that endomembrane system. The other thing is that, you know, the lysosomes are part of this endomembrane system, but this is really dealing with things coming in the other way, right? So we said there are lots of lysosomes in white blood cells, okay? So think of this as being a bacteria, okay, that this white blood cell is going to take in, takes it in via a vesicle again, fuses with a lysosome, okay? And this is also showing you where these enzymes are coming from, right? This is why the lysosomes are part of the endomembrane system. Anyway, those enzymes that are in the lysosome will then be able to break down that bacteria, okay? So all of this basically is that endomembrane system. All right, any questions about that? Right, so the second question asks about chloroplasts, right? And again, that's now topic of chapter 7 and chapter 8, photosynthesis and cellular respiration. So we will be talking more and more about the chloroplasts and the mitochondria. And I think I told you in the recorded lecture, for this exam, I'm not gonna ask you, you know, to label the parts or identify the parts of the chloroplast, because we talk about that when we talk about photosynthesis. So it's better to kind of talk about it in relationship to that, okay? So like, I'm not gonna show you a diagram and say, tell me what's the thylakoid, where's the, you know, the stroma or what have you. We'll do that in chapter 7. But in terms of functional chloroplasts, it's where photosynthesis takes place, right? Photosynthesis is basically you have two types of, or two main phases, right? Where energy is produced in one phase, that energy is then used to make carbohydrates in that other phase, okay? For mitochondria, cellular respiration is going to occur there, and that's where ATP is generated, okay? So, I mean, at this point, I'm just expecting you to know sort of just general function of these organelles, okay? So think of chloroplasts and mitochondria as the energy-related organelles. They're obviously not part of the endomembrane system. They're different. All right, so that's pretty much, you know, that basically concludes the review. So if you guys have questions, let me know and I can go over some more or clarify or whatever you guys need. But otherwise, I just basically reviewed all of those chapters 1 through 4. Again, remember that review sheet. If you have not submitted that, you can still submit it, okay? If the whole point is it needs to be submitted before you go taking the exam, because that's you reviewing and writing down the notes, okay? So remember, for that review sheet, again, you're completing it and then you're submitting it here where it says picture of completed exam 1 review sheet. Once exam 1 is available, you guys will click on here. You will have 90 minutes to complete that exam, okay? It is available. It'll be available from about 1 o'clock, might be a little bit later than that. I'll shoot you guys an email when it's available because I have to finish putting it up on Moodle. It'll be available around between 1 and 2, and then you have until the very end of the day on Friday to do it. Once you log in, that's it. You have to complete it, right? So 90 minutes at the time that you log in to complete that exam, okay? Any questions about that, guys? No? All right, so like I said, if you have not submitted your completed exam 1 review sheet, that's where it goes. Okay, I also already explained what I'm expecting on that. It's basically you adding your notes into that outline that's provided. All right, guys, if you don't have any other questions, then I'm going to end our meeting. You know, if something comes up, put it in the discussion forum, right? which is here, not the announcements, right? But the question and answer forum. Any question that comes up, best to put it there rather than email me. A couple of you guys kind of emailed, like you emailed about the review sheet assignment. Anything that pertains to assignments or anything like that, best to put it here because then everyone can see the answer, okay? And because then a lot of times I'll get the same question a couple times. So it just kind of helps everyone out. And even if you didn't ask the question, it might be helpful to you to see that answer. So try to remember to put questions there, okay? All right, guys, so that's it. Like I said, exam 1 will be available on Thursday. I will review the lab content and basically then you'll be ready to take that lab quiz that pertains to that. So all of your lab assignments need to be submitted by Thursday, right? By 11 a.m. on Thursday. So that is the lab metric system and microscope labs, and chemical composition of cells, and then the cell structure and function labs, okay? I wrote that in your email as well. All right, guys, so that's it. Like I said, unless you have any other questions, I'll stay on for a few minutes if anyone has individual questions. Thanks, guys. So let me stop the recording.
Thank you.
Thank you. Stop recording.